The life of a cell is cyclic. Um, a new cell develops, it does its job. Some point in time, it's going to receive signals from outside or inside of it, intracellular or intercellular, that state that it's time to divide. Cell divides, gives two new daughter cells. Cycle starts over again. Early biologists, early being, the last century and a half ago, when they were looking at cells under the microscope and they were developing new stains and things, they were studying cells. They were, cataloging, characterizing the different cell shapes, the different things they could see the cells doing. One of the things they noticed was that the cells divide. The cells seem to go through a series of steps before this division. They called this the mitotic phase because it was very dynamic. There was a lot going on here. They focused a lot of their time and research energy studying mitosis, this mitotic event where one cell splits, becomes two. They spent so much time on this, and they prioritized the study of this to the point that the rest of the cell cycle, when the cell's not going through this mitosis, they've just referred to this as interphase. It's the intervening time between the next mitotic event. Well, that's kind of weird, seeing as this interphase is when the cell's doing its job as a cell. Cardiac cells doing its job as part of your heart muscles. A columnar epithelial cell in your large intestine is doing its job in the tissues of your large intestine, absorbing water, doing all the things it's supposed to do. It's just during this mitosis event, this mitotic phase, when its job's put on pause. So, what we now focus on is the overall cycle. You look here, this figure, circle, as we've seen in previous chapters. When you see a circle for a pathway, a circle means cyclic. No beginning, no ending. Just this constant churning of things occurring. Stuff feeds in, stuff feeds out, but it's a constant, never ending until the cell's dead. Now when you look at it, part of it is in blue, part of the cycle is in red. The blue, this is the interphase. Notice that's the majority of the cycle is interphase. This is the cell being a cell, doing its job. That little bit at the top there you see up at the top, that's M phase, mitosis. Depending upon the cell, that could be as little as 10% or maybe as much as a quarter at most of the cell cycle. So let's focus first on this interphase. The time when the cell's being a cell. It's doing its job. It's interacting with its neighbors. It's reacting to cell signals that we talked about in chapter 9 that are coming along in the blood or in the interstitial fluid. What we found is that interphase itself is split up into 3 subphases. First, G1. G comes from gap. During this gap, nothing major is occurring, cells being a cell. First gap. At the end of the first gap, you go into what's referred to as S phase, synthesis phase. What's being synthesized? Well, this is at the time when all the chromosomes in your cell are being duplicated. You go from 46 chromosomes, 23 from mommy, 23 from daddy, to 92. Because remember, all those sister chromatids have now just been copied. Then, after S phase, you go into what's known as G2, second gap. Cells just being a cell. Now, during S phase, the cell is still being a cell. It's still doing its job. It's just that, a lot of its resources are going towards copying the chromosome. And when you look at the first gap, G1, this is immediately after mitosis. The daughter cells are smaller than what the normal cell would be, so during this time, the cell's increasing in size. It's starting to look like what a normal cell for that cell type would be, whatever it is. During this, cell's just doing its job. If it's a neuronal cell, it's being a neuron. If it's a muscle cell, it's being a muscle. S phase, as I said, this is when the sister chromatids are made. Every chromosome in that cell will be copied. An exact copy, the sister chromatid, will be made, and it will be kept attached to the original that it was made a copy from. We also start to see a spindle, the mitotic spindle, start to assemble. This is a structure made from microtubules that would, during the M phase, will be used to pull apart the sister chromatids. G2, cell now needs to replenish. Needs to go through and replenish all the ATP it just used. Needs to go through and prepare the cell for the very dynamic M phase. We also see the cytoskeleton. As G2 progresses, the cytoskeleton starts to disappear. You don't want all those cytoskeletal components getting in the way. You'll notice when you look here that there's that little circle attached, referred to as G0, gap 0. This gap 0, this G0, it's a, two steps, one step forward, two steps back kind of thing. As the cell is being its cell and it's progressing through G1, sometimes the cell just does not need to go through S phase. Either the cells around it are nice and healthy, there's no damage, or it's the type of cell that, it's never going to go through mitosis. If it's a cell that's in a nice healthy tissue, no damage, no reason to replenish, replace, basically this is turn around, go back, start over again. Turn around, go back, start over again. That's G0. If it's a cell that, it just is not going to go through mitosis, and, kind of like neurons. Neurons don't replace, per se. Well, it's going to hit that, and it's going to go back, and it's just going to go back, and it's just going to go back. They refer to this as being quiescent, inactive stage. The cells, their cells doing its job, it just does not need to progress through the rest of interphase.